Every living organism can become infected by viruses. This can actually be a good thing. For example, algae, free-floating plants in lakes and oceans, can form massive carpets of many kilometers in size. And for some of these alga species, these carpets or alga blooms are actually visible from space. As you can see here in the satellite image taken from the British coast. If this is a harmful alga, this would mean itchy skin after swimming and that the fish in aquaculture die. The bloom can be stopped when the alga is infected by a virus. However, before we can use any virus for control of the alga blooms, we need to have more information. For example, what kind of virus is it? How does its structure look like? And how does it self-assemble inside the alga host cell? Well, Czech Republic does not have access direct to the sea. So in order to address this structural biology question, we have to cultivate the alga in the lab. This alga is actually very small, about the size of a bacterium. Imagine that this pomelo is one alga cell, and the virus would be the size of this grape. And each infected alga cell can produce up to 25 new viruses. For more, there is no space. We know that because the cell is actually shaped like a lentil, and we can use a very strong microscope, such as an electron microscope, to look through this thin area of the cells and see the virus particles assemble inside. We can use the same microscope and study a pure sample of the virus. Here we take many close-up images of many virus particles. Each of them adopt a very random orientation. And then we use these images, which are two-dimensional projections, and reconstruct a three-dimensional model of the virus, just in the same way as you can tell how a person looks like, even if you only have photos from the side, from the top, from the front, and so on. So this three-dimensional reconstruction reveals all the components the virus is made of and the fine details on its surface, which are important for the interaction with the host cell. So overall, every structural characterized virus helps us to complete the knowledge of this huge diversity that exists in virus structures, assembly pathways, and infection processes. And it also helps us to understand why viruses are so successful in hijacking the world.